Well, welcome everyone to Mark Dane's thesis defense, and it's always fun to see people reach this stage. And I've enjoyed seeing Mark wend his way through the program and doing his thesis work in Joe Gray's group, and I understand that's going to continue after this, which is good. Mark has an undergraduate degree in electrical engineering that he obtained over 30 years ago, although he said he thought he might be the oldest person in the room, but he's not, unfortunately. Anyways, so we'll follow the usual procedure where he will give his presentation, and then the public, the audience here, can ask questions, and then the audience will be excused him with only his committee, and then he'll be excused from his committee, and they'll make the final determination of passing or not. So go ahead.
Is this picking up okay? Yeah. Thanks. Well, good morning, and thanks for coming. It's been 2 years plus to get here, so I'm really happy to be here to be able to present and defend my thesis. It's developing a computational pipeline for high-throughput quantitative phenotyping. And what I'll do is explain the background, the motivation for the motivation and background, the specific aims for the thesis, go through the computational pipeline that I developed and the data sets associated with developing and running those data sets on the pipeline, and then the major core parts of it, the exploratory data analysis, the normalization, and the scoring, and wrap up with lessons learned, next step, and certainly the thank yous and acknowledgments for how I've gotten here. The motivation, I mean, for why I am here is it involves things like the National Cancer Institute estimating 41% lifetime risk of cancer, just staggering numbers. 1 out of 7 men getting prostate, 1 out of 8 women getting breast cancer. And so if it doesn't affect us directly, it certainly affects our family, friends, neighbors, coworkers, and such. And so trying to be a part of figuring out how to solve that's a very high motivation for me and for the people that are around me and people involved in the field. And one of the things that we've focused on is using cell lines to be able to to be able to predict the responses of cancers and to be able to do our experimentation. And that's proven to be very effective. Starting about, I think it's almost 25 years ago, the National Cancer Institute started developing 60 cell lines, cancer cell lines, immortalized cell lines. And since then, they've been involved in developing or discovering over 45 anti-cancer drugs. So it's a proven method of using the cell lines to be able to develop anti-cancer drugs, which is ultimately our goal in the Gray Lab. And the other part of this is that cell lines do recapitulate what happens in tumors. They recapitulate the subtypes and the features. And so that's very important for us to be able to use them as effective models. One of the things that we would love to do, this is data from 18 years ago, Brian Drucker's lab, where we've got cell viability and days, up to, I believe it's 4 days. And we're looking at cell lines that have the Philadelphia chromosome that causes chronic myeloid leukemia. And we're looking at cell lines that have been treated with imatinib or Gleevec, or untreated. And what we see is that the controls and the untreated cells continue to proliferate while the treated ones die off. And so it's signals like this that lead to beautiful data like this, where 10 years later they were able to publish that over 5 years out that over 90% of the people were event-free, meaning they did not have recurrence of the symptoms of CML when they were taking imatinib. And so these types of signals, unfortunately, have been very, very difficult to follow and are to find again and to track through. And so what we've just found is that we need to take a more complicated, a more complex view on this problem. And so this is a functional genomics approach where we're perturbing the cells, these cell lines, and then measuring many different aspects of them. And here's an example of some of the different aspects. We're using immunofluorescent imaging that we can tag different molecules, different proteins in the cells, and measure and quantitate and measure how much they are in individual cells, while we perturb those different populations in different ways that lead to hallmarks of cancer like apoptosis, proliferation, differentiation. And so this type of data that we develop on both a population and a cell level gets very complex very quickly. And so let me step back from that. Let me talk a little bit about these perturbations that we expose the cells to. We're using really two major kinds. The first one is short interrupting RNAs that are involved in gene silencing. And they have the ability to silence specific genes within a cell, and then we can look at the phenotype. And a quick run through is the siRNAs come into the cell. They become part of a RNA-induced silencing complex, and they make that complex very specific to cleaving mature RNA sequences. And so these the transcripts. And so by perturbing, by exposing the cells to particular siRNAs, we're able to see the effect of silencing one gene at a time. The other perturbation that we use is by modifying the cell's microenvironment. And there's been a lot of work and discussion about how cells are affected by their microenvironment. And these involve signaling proteins, structural proteins, scaffolding that results in the cells acting different and differentiating. And there's hypotheses that this microenvironment has an effect on cell drug resistance and differentiation. And so we want to be able to expose cells to different microenvironments in a controlled fashion and measure the phenotypes. Over all of these, we also have the ability to expose to drugs and see and check for interactions between one or more drugs within any of these environments. This kind of work has been done for several years, for quite a while actually, in well plates, and it's a standardized, it's a well quantified process, but it has some. And so part of our motivation, though, is to miniaturize that. The work in the Gray Lab is miniaturizing these into microarrays. And part of the motivation of that is both in cost improvements. So some similar microarrays have proven to be 15 to 30 times less expensive. They're much smaller. They're easier to handle. They don't require the kind of dedicated core facilities that large well plate experiments require. And there's something that we can do with I don't want to say a small amount. We have bought in the neighborhood of $500,000 of specialized equipment to be able to create these microarrays, but it's not in the level of the core facilities. These microarrays give us the ability to do many more experiments, to look at many more endpoints, to be able to look at many more perturbations in cell lines. But that also creates a great deal of information, a great deal of data, and creates the need for the automated pipeline that's at the core of my thesis project. Two more slides on introduction to be able to introduce some of the artifacts that go into this data set in order to be able to make sense, to be able to talk about how we then locate it and remove it. In manufacturing these microarrays, whether it's the microenvironment ones or the siRNAs, they take a similar process that really start here with these pinheads. And here's a blow-up of the pinhead, 4 by 12. So we have 48 pins. And we dip them into master library plates that would have whatever perturbation we want to expose them to, and pin spot them onto plastic arrays that have been treated. Now, this is a process that's very similar to cDNA microarrays that have been done in the 90s and such. And there's a lot of statistical evaluations, a lot of methodologies that have been developed that I've used to be able to do some of the normalization and scoring and analysis in this pipeline. The arrays, one of the things that at the printing level, it's important to realize that, say, one pin is confined to one pin grid. So all of in this case, 81 spots at this level were made by pin 1, and pin 2 made all of these in pin 3, and that becomes important down the road when we look at normalization. But after we move on past the spotting, we're now doing things that are at a global level. We flood the array with cells. They adhere to the spots. We wash them off. They stick only to the spots. The cells grow exposed to whatever perturbation is there for typically around 2 days. Then they're fixed, stained, and permeabilized, and then we do the measurement. So these types of activities cause perturbations, technical artifacts at the global level, and we'll see some of those. And the others, these pins, cause things at this pin grid level. After the staining, the cells are then imaged, and we have 2 different types of systems that we image on. The TCAN is a very high-speed, high-throughput system. It has 3 lasers. Does not have DAPI, which is significant to us because DAPI is a great way to be able to locate cells, locate their nucleus, the DNA in the nucleus, and it's missing from this machine. But these are the kind of images that we get from within a few minutes we get from the TCAN. On the high content side, with the ScanR is a high throughput, it's an automated microscope that does 4-color imaging, high resolution, fully automated, where we can actually make not only the measurements of DAPI and the other color intensities, but we can make measurements on the cells, the morphology, the area, the perimeter, and get accurate cell counts. And so the pipeline accepts data from either one of these, but what we also learned is that these types of things bring in technical artifacts as well, the data that needs to be taken into account. So my specific aims have been to these 3, to be able to develop the pipeline that does the EDA, the normalization, the scoring of this data, develop a method to be able to simulate these data sets because it helps to be able to optimize and characterize the pipeline and to develop it, and then to apply that to the simulated and some actual data sets and analyze its performance. So stepping into the next part of the talk, actually exposing, talking about the computational pipeline. And so I started on this about 15 months ago. And much of this was very, very new to me. And I would say that we're looking at a snapshot in time. It is something that will continue to be developed and worked on. But it's already proven effective in a lot of the work in the lab over the last 9 months especially. So it has these major sections. No bioinformatic project can start without cleaning data, and this certainly has to happen. We're taking data from the printer, from either the TCAN or the ScanR, and then we're also taking experimental data. Those files need to be merged so that they're all in line. The data is merged with the metadata, and then from there, everything, the round circles represent software objects. And this was all developed in R. And so these are R S4 objects. And the advantage of that is that they're structurally very sound. There are R bioconductor packages that take this format and do additional operations. The core of this is a bioconductor package called Cell HTS2 that was designed for doing well plate, doing experiments like this that are based on well plates. And that's great. It provided a backbone to do things from start to finish, but there's significant differences in doing microarrays versus doing the well plates. And so part of the work that I've done is to bring in the pin grid arrays into the well plate type of analysis, and to be able to bring in more work on multiple channels, which the Cell HTS2 had facilities for that, but it wasn't very well developed. And then also at the back end, our data has many more channels, and so scoring for that is a challenge. And so some of the methods there, I implemented some methods that do a more robust scoring methodology. So that's the first specific aim, and we've been able to run this. I should also speak to one of the other big goals of this is not only for it to be extensible by using these software objects and using the bioconductor code, it allows me to build on other people's work and also to be able to make it modular so that I can, as I optimize things, I can replace the different modules. But it's also reproducibility is just a vital part of experiments and computational pipelines like this. And so at the end, these objects get the information get converted into HTML files, into reports. The whole scripts get connected and stored. And so it's relatively straightforward to be able to go back to the input data and get the same results, and to have the process documented all along the way. The second aim is to create these simulated data sets. And this has been an important function to be able to not only fully develop and test the pipeline, but to optimize some of the processes. And so the way this process works is through the, first of all, creating these sample values. The sample value is the end result, but the base values here are sampled from a gamma distribution. And I chose that because it does a good job of emulating the intensities that come from the scan-R and from the TCAN, where they start at 0, go forward, have mostly, I've chose the shaping of it so that it emulates it having a large grouping of distribution and then kind of a long tail, which is typical of what we see in this data. Those are then modified by a factors that represent the pin grid factors that perturb that data separately for each pin grid. And so that's where we get this checkerboard effect, where we've chosen from a normal distribution that's centered around 1 to be able to make minor modifications, and that can all be modified as well. From there, a similar effect is done to be able to create replicates, but here every location gets a sampling from a normal distribution, so that each replicate comes out just a little bit different from sort of the base plate, the master plate from that. And then at the end, I'll show where I do some spatial perturbations to simulate some of those global perturbations that we see in actual data sets. In addition to that, with the simulated data set, I can create controls and I can create hits. And that's kind of the beauty of that. It's not really cheating. You actually get to create that, but it allows to be able to optimize and quantify the pipeline. And so here, it's a little bit hard to see, but there's a series of blue lower intensity that are the control values and then the hits. And so these control values, I set at a 70% reduction. The hits, I set at a 40% reduction. And we'll look to see how well we can find those after the normalization and scoring. Here's some sample data sets that have come through the process. And then here are the two simulated replicate that I did the analysis on. And so you can see that in addition to these sort of global, what I call global or regional effects, here and the hotspot here and lower intensity here, hotspots, these also show some pin grid where some of these intensities are more focused on the pin grids. And as we started this process and as I was doing the development, we started seeing pin grid issues in the coming from the printer. And so initially I put a lot more focus on being able to normalize at the pin grid level. Since then, we kicked the machine out, we bought a new one, and it looks like the pin grid issues are not as significant. But we're still seeing these spatial, these global issues that we associate with things like staining or washing or cell seeding that's non-uniform. So here, in addition to the pin grids that are obvious here, and this has pin grids as well, these pseudo images show an area of ellipsoidal images at varying height that's higher intensity and one at lower intensity, and then some linear perturbations. So these can be located anywhere within a microarray and within the replicates. And so that's part of the simulation that I've done, is to be able to create different sets to modify or to emulate what we see coming into the lab. Two other data sets that I'll do some processing are an aberration data set here, which is an siRNA-based data set. And it has 4 different channels of DNA. This is DAPI. So this is the amount of DNA in each cell. These are byproducts of apoptosis. This is DNA repair, and then this escapes me, and I don't want to say it wrong. But it's another endpoint marker. And so the pseudo images are part of the EDA that's done, and what I've found in doing these EDA is that there's not one graph that shows you everything you want to see. You kind of get maybe 2 or 3 of the things you'd like to see. Here, the pseudo images are of the intensities, And so we can see where things are higher or lower. So we can find these effects based on the regions where they're at. But they don't really show a good job of intensity because they're auto-scaled. And if you eliminate the auto-scaling, you can get some better idea of intensity, but then you lose where the effects are. Whereas histograms do a good job of showing the intensities and how things are distributed, but then you lose, where are these peaks in here? So you're really looking at these side by side is very important. And then box plots are another tool that I'll bring in that helps. So the other data set is kind of running the other way because this has 4 replicates of one channel. And this channel is a cell count channel. And these are microenvironment microarrays. So here we can see that there's sort of some funny patterns that are associated with pin grids in these rows here, and there's some down here. And then there's a column here that has higher intensities than the rest. And that's seen at some level through all of these. And that becomes significant as we go through the EDA. Yeah, grab some water. So a quick summary of these data sets that are run through the pipeline. The aberration, and I've chosen these columns because as it turns, these are some of the controlling features that come into the experiment design. So having randomization is very important in these types of experiments, and I'll get into some of the specifics on that. The aberration screen is fully randomized. The MEA data set is not, and the simulated one is. We have replicates as an important feature to be able to pull out the biological signal over the technical noise. The replicates, there are no replicates in the aberration. We have 4 in the MEA, and there are 2 replicates in the simulation. And lastly, controls. There are no positive controls in the aberration screen. There are no controls in the MEA, and then there are controls and actually simulated hits in the simulated data set. So going through the pipeline with these data sets is what I want to do now with the EDA, the normalize, and scoring. One of the things that became obvious to us as we were working on other data sets that are similar, we had a data set that had 400, that resulted in almost 400 well-played pseudo images. And we wanted to analyze that for spatial variations. And pseudo images are great for showing you where things are, but when you're looking at 400 of them, it's hard to prioritize them and to be able to know which ones are, you should focus, and potentially which ones the experiment may have issues and may need to be redone. And when I did some research, I didn't find any quantifications that did exactly what we were looking for by index or by valuing, quantifying the spatial variation. And so I developed a method that I did spatial variation index. And what this does is fits a bivariate local regression to the data. And so there we've got basically I think of it as sort of like a smooth contour map of the data. And what we want to do is we want to pick up on these regions of spatial or whether they're pin grid or global variations, but we don't want to pick up on the actual hits or controls. And so these isolated points, that I call isolated, the bivariate local regression is tuned to be able to ignore that. And then we use that model and measure all of its variations from a flat median surface and then normalize that for the size of the array. And so this results in a single number that captures how much variation there is in the array. So an array that only has isolated points will come up with a very low value, and one with high spatial variation will have a higher value. Some of the initial results that I did on this look very promising, and so it certainly warrants more work and more validation. But when we look at these, here these pseudo images are all scaled the same, so you can see that the variation is getting more and more intense, these side variations. And then those ellipsoidal variations, here we're looking at the spatial variation index going from 0 to 0.3, and then as the perturbations are normalized to go from 0 to 1. Here we see that as these variations as the perturbations increase, the variation index tracks accordingly. The same thing for these pin grids. As the pin grid perturbations increase, the spatial variation index increases along with it. But on the isolated one, as the isolated variations increase, the spatial variation index does not. So this is achieving at an onset of what I was hoping for, of something that captures that variation. As we were going through the data set, this MEA data set, we needed to look into why these effects, what was the cause of these effects, of these localized intensities and lower and higher values. And so one of the first things I did was color code the ECM proteins and the growth factors. And the colors don't mean anything except that I wanted to create as much contrast within a color set as possible. Made them transparent and then overlaid them on each other to be able to see where things were. And so the way these MEA arrays are created is it's every possible combination of these 16 ECM factors and of these 24 growth factors. So that creates 384 different combinations. And so this plate has 6 replicates of 384 combinations in it. And so here we start to see that there's something kind of funny going on. And then when we break apart the ECM proteins from the growth factor, we see this very structured location of growth factors and ECMs. And so what we determined was that because these growth factors, the biology and the locations were totally confounded. And that became one of a series of issues with this data set that stopped us from taking it further in analysis. So even though we're seeing effects that are likely associated with the biology, they're also likely associated with the location, and we don't have tools to be able to pull those apart. In order to be able to fix that, it's not trivial, and this isn't necessarily part of the pipeline, but in doing these explorations and finding what the problems are, then you have to be kind of part of the solution. And what we found in discussing this is that all of the proteins were in a row, all the growth factors were in a column, and so when we do the pin grid, the pin spotting, we have 8 pickup points within this master library plate. So we go here to 1, and then we go to spot. So the pins only were able to have access to a small subset of the perturbations. And then the plate was actually halfway through. The plate was turned halfway around. And so that's why, if I can get back, come on, I confused it. We see this, the turning of it halfway around created the top half to be similar to the bottom and the left side to be similar to the right side. So it helps, but it's not what we needed to do. What we need to do is, if we could create 10 different master plates that have 384 master plates, then the entire microarray can be completely randomized. And we can do that with siRNA. It's just the nature of them. They don't have these families like you have in the microenvironment, in these growth factors and such. But in order to be able to do it with the MEA arrays, we've developed a methodology that we haven't implemented yet, but we're kind of working through. And so it starts with the growth factors and proteins. We're going to plate them into 96 well plates, put that onto a machine that can pipette individually from anywhere to anywhere, and then do a mass transfer from 96 well plates that are fully randomized into 384 well plates. And meanwhile, tracking the data, what goes where throughout this whole process. It's not, the idea would be that we put everything that we want to place, we have everything where we want to go, and we can pick from anywhere and go to anywhere, and then we get 100% randomization. That's not what this is going to create. And so we need to be able to be careful, and I want to simulate this to be able to make sure that when we're done, we do create enough randomization that we don't have anything like that structure that we had before. And so what I'll be doing is going through that and then feeding back and determining what should happen throughout this process so that we do get to randomization once we're actually at the printed microarray level. One of the other tools that we use in the design and the experiment design are replicates. And the replicates are very important to be able to increase that biological signal. And so the typical way of looking at the replicates, the results, is to be able to plot one replicate against the other in a scatter plot, the intensities. And the ideal replication would be everything would be on this diagonal line. And then we would probably do a Pearson correlation and hope for a very high correlation. But in these experiments, there's sort of we're very concerned about the tails. What's going to go forward in these, once we've got valid data, is what is in the tails of the distributions. And things like Pearson correlations are dominated by the majority of the data points, which to us are going to be non-reactive samples. And so there's the tools that are often used here are not as useful here. But one of the things that the replication analysis does let us do, and this is the simulated data set, it lets us kind of confirm in another way that something is going on that looks, when things are off the diagonal, we ought to know why that's happening. And in this case, there was in the simulated data set, there are intensities that are much lower in just one of the replicates, in replicate 1. And that's why these data points are pulled off of the diagonal. And so it's kind of confirming to be able to see things at this level. The other thing we can do with replicates is we can do coefficient of variation calculations. And so that allows us to be able to see some idea of how reproducible the biology is. But the coefficient of variation is not a very stable statistic in some situations, especially when we get down to a low number of replicates. And so what I wanted to do was check the null hypothesis of that. So I created a data set of totally random results, normally distributed, and pulled, but two different distributions. One with a standard deviation of, with a standard deviation 10, and the other with a standard deviation of 20. And then pulled 8 replicates out of each distribution. Did that 1,000 times, and calculated CVs on that. And so there's really no correlation between any of these. And I just wanted to see what the distribution of the CVs are, what the null hypothesis would look like. And if I were to set a 5% threshold on that, what would that threshold be? And what you can see is that the threshold comes out at different levels. And what this is telling me is that using CVs depends on what your data is and what the distribution of your data is. And so there's not a hard and fast threshold that we can use to say, yes, that's a good one, or no, it's not. But the replicates, this isn't to say that, replicates are still very vital, and we need to have replicates in our experiment to be able to increase the biological signal. One of the things that comes out of this is that it's easy for us physically to create replicates within a pin grid, but they're not as valuable because they're that same effect of, like we saw before, then they wind up, the biology is confounded with the location. After the EDA, one of the things I should say is that we take this raw data object, we do the EDA on it, and we then pass it through to be able to normalize it. But also, after we've normalized it, we take a normalized version of that and do more EDA on it. So the EDA is used on both the raw and the normalized data. So part of what we need to do when is to normalize and normalize. There's a lot of different normalization methods, and we want to choose one that is effective or that matches the technical variations. Because ultimately what we're trying to do is separate the biological signal from the technical artifacts, from the technical variations, the technical noise. And so what I'm showing here is that a common way to do that is to simply divide by the plate or the well or the microarray median. And that allows you to compare microarrays one to another. But it doesn't do anything about the spatial artifacts, the regional event, any of the artifacts. It just all centers them at the same location. And for our type of data, at least what we've been seeing so far, we need to deal with these regional artifacts. And so there are spatial normalizations that go back to the one that I've used the most has been to go back to that bivariate local regression, that smooth contour map, and use it to be able to do a normalization of the sample values. And so starting with these two replicates, this one having pin grid effects, this one having pin grid and these spatial normalizations. Applying a pin grid normalization to it really does a very good job of bringing all the pin grids up to the same level, and now the variations can be compared across the microarray. But on the one that has these global effects, these are not pin grid effects, and raising each individual pin grid doesn't solve that, doesn't remove that technical artifact. And in fact, one of the things you can see down here is we've actually distorted this data pretty significantly by forcing something that was very uniform and low intensity to have a median that's the same level as the rest. And so that is one of the, we talk about normalizations wiping out or covering over the biology. That would be an example of it. But by applying this spatial normalization at a global level, we wind up with microarrays that are virtually free of technical artifacts, and that this should now be the signal, the biological signal. You can still make out here the effects of this line, but for the most part, that the technical artifacts have been removed. And I should say, I mean, it's one of the areas that we want to tread lightly, and I think of doing the least amount of normalization as we can, and to be able to do valid comparisons and not want to do any more, because we can make them perfectly smooth and beautiful data that has no biology left in it, which would not make it beautiful data. So the spatial normalization has one parameter that overrides, or is a very strong parameter that I use to optimize it for this process. And that's the NN parameter. It's a number of neighbors. It determines how the weights, the spatial parameters, spatial normalization does a weighting of the neighborhood. And so you're, in essence, determining how far out that weighting should be. So for instance, if at a value of 1, it includes everything equally weighted. So it's the same as a median normalization. And at very, very tiny values, it basically takes the data point itself, or very, very localized areas around that data point, very few neighbors. So part of what I needed to do was to choose where in between those two extremes the normalization value should be set. And these are partial ROC curves where we've got sensitivity and specificity. And what you can see is that this is another sort of thing that happens with these data sets, is that there's so many values that are negatives that they dominate these curves. And so it's hard to get valuable information out of doing these ROC analysis, because everything is up and to the right, because the data set, even by saying everything is a negative, well, it mostly is. So you wind up with curves that come up very far to the right. But what I did was set the NN to different values, trying to get a sense of what might be an optimal value. And then using the values at different, here I'm using the positive predictive rate and plotting it against NN values and picking thresholds. This we have to be able to get a positive predictive rate. We need to have a threshold that says everything below this is a positive, everything above it is a negative. So to be able to pick that threshold, I use the ROC curves and pick the values that are closest to the upper left, and then calculated the positive predictive rate at different NN values. And so this gives me a sense of what sort of positive predictive rates to pick. And the thing that I liked about the positive predictive rate, or positive predictive value, is that what it's doing is it's saying, given a hit list, given what we call the hits, how many of those are true positives, how many are false positives. And so that's a really effective tool to be able to give the biologist down the stream to say, here's a hit list, and I can give you some idea. Is it 10% valid hits and 90% false positives? Is it 50-50? It's nice to be able to give some sense of what the level of false positives are in that hit list. And so, but also in doing any of this kind of work, there's a concern of not only over normalizing, but over fitting to the data. So I picked values that are in this range to be able to go forward. And this is another area that I may need to come back to as we get more and more data, but I feel like I've got the tools to be able to pick these values and to quantify the end results. And then applying that to the normalization, or to these data sets, the aberration data set, here's the raw data, and you can see these pin blocks. And in general, there's a trend down in this one, which is because they start out brighter and they go darker. Here we've got this lower intensity, higher, and then lower intensity that shows up in these pin blocks, in the pin grid box plots as going up and then down in these series of 4. We go up and down, up and down. And so this is actually a way of being able to quantify the raw data as well as the same kind of graphs on the normalized data. Everything is a much tighter distribution, and yet we still have information in the tails of the distribution. And even though we didn't force each pin block to have a median 0, they all come out with very similar medians. And so to me, this is a way of qualifying or evaluating the normalization and determining whether it's kind of gone too far or whether it's doing a good job. The other way of doing a quality assessment is by using the controls. And so in the simulated data set, this is the kind of this is what we would love to be able to see in all data sets. But the result, I said I'd knock those controls down by 70% and the hits down by 40%. And so after normalization, controls wind up well to the left in the intensity of the body of the data. And the controls, these purple ones in here, are to the left. They're still a little mixed in with the samples. And then our negative controls, where there's no effect, are used to be able to determine what the most non-reactive samples are doing. In the well plate world, a typical quality assessment is a Z prime factor. And what this does is give you a sense of how separated these two distributions are of the positive controls and the negative controls. And in there's quantified values. Z primes less than 0 mean that it's a low quality plate. Between 0.5 means it's a good one. Between 0.5 and 1 is an excellent quality plate. But we don't have these kinds of numbers for microarrays. And in the published microarray for these cell spot and similar cellular microarrays, I haven't seen anybody publish a Z prime factor, even though they typically come from the same well plate world. So they talk more about separation. So, I know there's a challenge there, but we still need to, I think there's a lot of value in carrying controls and control analysis forward in these microarray experiments. So moving on to the scoring, the idea of scoring is to be able to create a prioritized list of hits that are going to be validated in the next series of experiments. And sometimes those experiments are not very resource intensive, and sometimes they are. So we have to take that into account as to how big that list can be. Can it be a quarter of the data set? Can be, if we had started with 4,000, do we want to winnow it down to 1,000, or do we want to winnow it down to just a few hundred? So that's important to take into account. And the other thing that I in doing literature search is I came across the talk of using ranked products versus Z-scores. And this was very appealing to me because one of our challenges, we have many different channels, many different endpoints, and we have different cell lines. The beauty of the technology, the miniaturization, is that we can create these large data sets. But then when we come down to the end, we need to be able to compare them in ways that make sense. And the way that I think of the ranked products was proven in some of the analysis to be able to be a very robust way of doing comparisons. It's relatively simple to calculate. It's basically you have a list that can come of perturbations, and then you multiply. It's the product of the ranks across these lists, and those follow a gamma distribution. And so we can easily determine how likely a particular rank product is for a given experiment. And so that allows us to set thresholds on things like p-values as opposed to just quantifying, saying, at a certain point in the distribution, or it's a repeatable way of setting a threshold. So I wanted to do some analysis of, in our situation, on the simulated data set, what the performance of ranked products is. And so here we've got, again, the positive predictive value versus list size. So this is the hit list size that we want to go downstream. And in this case, there were about 143 controls and hits that are true positives, and all the rest are false positives or negatives. And the way to interpret this is we've got 3 different levels of normalization done to the data, and then in blue the results of doing z-scoring versus the results in purple of doing ranked product scoring. And what you can see is at lower, this level here is a distribution of the incoming data that has not been normalized. So it's it's not normalized data. As we get higher, the distribution gets more and more normal. And there's actually a level, it's hard to see in this, but there's a blue line that when the data has been well normalized, when it comes from a normalized distribution, z-scores perform very well. In fact, they slightly outperform the ranked products. But where we're operating in these range, the ranked products turn out to be very robust and non-normalized data. And what's happening here is we have two replicates. One of them resulted in a z-score that was very extreme. And when we average those, we still get a z-score that is well outside the rest of them. And that doesn't happen in ranked products because it doesn't try to determine how far extreme it is. It just orders the list. And so I think it's turned out to be a very robust method. And I think it's a way that allows us to do a little bit less normalization, which is of value to make sure we don't wipe out biological signal and still get a robust result. Here I've picked one level of normalization and looked at the ranked products versus the scoring. And because the level of the data coming in here was pretty normal, z-scores do a good job. There's one point in the normalization of that data where the ranked products overcome the z-scores. But I still want to do a little bit more work here to be able to understand some of the implications. But so far, the takeaway is that the ranked products seem to provide a good job of being robust predictor. So last few slides, we've learned a lot from applying this pipeline. First about the randomization, the importance of that, and uncovering and looking for that. And we're going to go to great expense in order to create that randomization because it needs to be there. Trying to be able to do different normalizations and match the normalization to the artifacts. And so the EDA really helps helps us figure out what the artifacts are and then can allow us to evaluate the normalization. Using replicates in these and some of the initial data sets that were not replicates, we need to include that in the design. Using controls, we didn't have controls in some of the initial data sets. Those need to be a part of it to be able to do this assessment and to evaluate the normalization. One of the things that I didn't get into is we had signal-to-noise issues. And so that came out through the EDA. We tracked that through to its source, and what we found was that it's really critical. And in this case, there was a mismatch between the dyes that were being used and the excitation of frequencies that were on the equipment and that we were measuring at. So that all needs to be matched really tightly. Some of the next steps that I'd like to do is, I didn't mention, but along the way I had a chance to talk with Terry Speed, who developed some of those cDNA, the spotting mechanisms, especially the lowest pin grid things. And when I had a conversation with him, and he had suggested some alternate, more modern ways of doing normalization. And where I wasn't able to complete those as part of the thesis, there are things that I'd like to be able to add to this, and Terry offered to help with that. I think the scoring on here could really benefit from doing a clustering approach. We're in the process of doing this analysis at the cell level, and so rather that each of these populations, the data I've been working with here represents a population of maybe 500 to 1,000 cells, and or I should say, 50 to 200 cells, and we have the ability, and we started to do analysis at the cell level. These three all involve the fact that it's a work in progress, and so testing, user documentation, user training. Ultimately, I'd love to be able to make this into a package, but I've got a ways to go in that realm. Boy, it's great to be able to be here at this point. I want to thank Shannon for all that over the last 2 years, bringing me to this point, pushing sometimes, pulling many times, and always being there to help me out and understand how to get this far. Laura has been mentoring me in the Gray Lab and has been invaluable in helping me focus on what's right, supporting me when I've slipped, and helping me get through to actually being a research associate starting in a few weeks, which is very exciting to me. Z has been instrumental, along with Yuha, Nick Wang, Jim Corcola, Spencer and Sophia is all on the biological side of helping me understand, well, what is going on? It's not my background, and I had tons of questions, and so that's been very important to be able to understand the biology and all of the staining and imaging to be able to then bring that into the pipeline. And Joe has been instrumental. He's, for one, created the opportunity, but then also backed up the analysis when it showed things that caused experiments to be rerun, and that's felt good as well. In my life, my wife, Marty, is, she's the one who's been there all along the way, had to take care of our kids, feed me when I didn't have time to do what was right, and so she doesn't get the degree, but she certainly has earned it. And so with that, I've left a few minutes for questions. Thanks. Yeah.
Talk.
Thanks.
Can you talk a little bit more about the ranked product score? Does it give you some sort of statistical p-value? You kind of mentioned that, but then you showed the rankings, and I was wondering what happens if you rank a bunch of things where there is no true positives. Do you still wind up with a top hit list, and so then how do you interpret that?
So what we're doing there, I mean, if you imagine a list, let's say from 1 to 10, we've got 5 lists that are all 1 to 10, and they're the same perturbation. And so if that perturbation is 1 on this list and 5 and 5 and 2, multiply those numbers together. Well, when you do that, anytime it follows a gamma distribution where there's a high peak and then a long tail, and we know we can pick off the p-values for that distribution. And so it allows you to say how likely is it, given that p-value, how likely is it for a perturbation to be at those particular locations on that set of lists? Does that?
Yeah. So then what do you do with those p-values?
Well, what I want to do with them, what I started experimenting with, is say, well, what happens if I set a threshold of 0.02 and say I'm going to take every perturbation that has a p-value greater or less than 0.02. So because those are just those are in the tail, those are unlikely. And that becomes my hit list. And so, when I looked at 0.02 with the simulated data set, it did a great job of capturing the true positives and not too many false negatives or false positives.
So you're thinking about doing anything with like false discovery rate or anything like that, so you could kind of scale the amount of false hits in proportion to how many hits somebody's really looking for or something like that?
I think that makes sense. I sort of been waiting to see, well, we have a number of what's going to happen in the lab and what the tools are needed in the lab. So, in a way, I'm kind of waiting to see if we're going to say how we want to establish and what our capacity is for the next process.
A number of places in your talk, Aaron just brought up one of them, and then with the spatial index and the CD and the ranked product, you keep talking about a threshold. And I wonder if you could comment on some of the dangers of threshold in this type of workflow, and in particular, the types of things that would make you more wary of giving members of the lab a firm threshold to look for.
Yeah, well, I think that's a good point because, calling it a threshold sounds like it's a defined, hard limit that's really discerning what is and what isn't, what should be looked at and what shouldn't. And that's not, it'll be important as I speak because it's not how I see things. And setting these thresholds in a way it's not arbitrary, but there's many different ways of setting the thresholds and of deciding of what that means. And so I think I want to be careful about things like overfit. I want to be careful about ranking things as opposed to putting hard lines. But I guess in the end of the day, I want to see, well, what is our capacity downstream for things like hit lists and try to set a threshold appropriately. So
this also speaks, though, before the prioritization just to the EDA step. And in particular there's a real need to not lose track of the data, right? Especially the visualization of the data.
Yes. Come on.
This is Jim Corpolo's data, right?
One of the MEA Dataset is Sophie Bornstein's data that she did using Jim's microenvironment arrays. But I don't know if it's a different setup, a different layout than exactly what Jim had.
Oh, I see. So in your high throughput pipeline, this is probably just categorical nitpicking, but you place actually an EDA piece, right?
Yes.
That doesn't really imply somebody visualizing or looking at things or scatter plots or, right? Because, you would do EDA, but then whatever you think that seems useful, you include in your pipeline. So technically a pipeline, you wouldn't say a pipeline has an EDA component. Again, this is just it doesn't change what you're doing at all.
Okay.
It's just a naming thing.
Okay.
So actually, I completely disagree, because EDA is critical in a pipeline, and it is a step in which there is a human component. So the EDA can be generated automatically, but you still need inspection, and it can alter. So I think having an EDA step in a pipeline is actually fundamental, and when it's not there is where things go wrong.
Yeah, well, that's exactly what I was asking. So if you're actually, if this is involving a human looking at it all the time, then actually that's perfectly fine. I didn't realize that. Because usually when you say a high throughput pipeline, it's just assumed that this is running automatically and not sort of somebody sort of making decisions, first of all, visualizing something and then going and, let me look at something else. If such a component is there actually, if a human is involved in making decisions in running, that's fine. But then it's probably, then I guess it's hard to call it a high throughput pipeline, right? It's
just a hybrid, right? So you can still do things in bulk, but there is an inspection step. And that's not uncommon, actually, from many of the pipelines that are out there. Now, whether or not people choose to look at that is, an individual decision, but it is actually really common.
So is that kind of a philosophical thing where you could actually have all this in a true high throughput pipeline where everything's generated in bulk, but philosophically you're not supposed to look at anything until you look at the at the QA and the visualization up front, and if it looks fine, then you can look at the other stuff. But if it doesn't look fine, you should throw that stuff out without looking at it, kind of like statistical testing, right?
Or not even go that far. Automation can happen in batch, right? So things could come off the machine. Everything could be initially processed. The QA could be done, sitting there. And then there is an intervention step where you could continue on or you could check it.
You could. I'm just thinking about true high throughput where you want to keep things running, and you might not want to, if you really were doing this continuously, getting back in the queue might be sort of disruptive. So if stuff usually works and there's sort of a flow that needs to be verified, but is usually dependable, you could do the whole thing. But I just was wondering how much of, for scientific integrity, how much of there is this draw to look at the end results and not actually go through all the steps because they're sitting there on the computer somewhere.
What I've experienced is, this is a developing technology, and while it is, and until the results are, highly repeatable, then the EDA becomes a very integral part of the pipeline. I understand, wanting to jump to the hit list, but there's so much information in this EDA that it actually, I put more of my energy into that than I expected because of the maturity.
So as you see this being deployed, at least initially, is there this sort of open person in the loop idea where the EDA is generated and then they have to do something to submit the work for the rest of the pipeline?
Yes. Yes.
There's one other thought to that, too, which I'm just going to comment on. So the idea, especially for a lot of these metrics, is that you could develop flags such that the EDAs run, and if none of the flags are set off, then it continues on. But everything is still generated for inspection if you want. Whereas if flags are set, it comes to a halt, as it should, and then you have the ability to look at all of those plots and do the inspection. So the real issue is how high throughput do we get and lose track of the data, which is the real danger. And there is an ethical issue there, right? If we just keep going through and running all these analyses versus really being able to have the time to make sure that everything's okay.
And I mean, one thing sort of I've done with Jim's data is it's similar data, I suppose. It's not the same data.
Yeah,
if you actually try to, right now, I mean, this is a great normalization for doing the spatial thing, but it does everything in a smooth way. It sort of spreads everything, right? So, but if you actually try to detect batch effects, like singularly, using something like a surrogate variable analysis, where the variables are interested only if they merit to be so, then that would be a good comparison for you to definitely to, because some of the big effects may actually trigger some, sort of drastic sort of normalizations, whereas you can leave a lot of the sort of the other sort of biologically interesting areas alone that way. That's sort of a more of a not normalizing in a like across the whole variation, but really trying to pick a few events which may have led to the dependencies, which should not be there. So it would be a good comparison in your to-do list.
I agree, and it's one of my next steps, and I started it, and I realized there's not enough time between when I started and getting here today to include it. So, but I agree. Yeah.
But the one caveat, which is I want to say because this is being recorded, is that with SVA, if you do not know all the sources of variation, or in particular, you're not clear about all the complex signal, you can remove the biology. So you have to use SVA with extreme caution, whereas there are other methods where when you know you have a batch effect potentially, like Combat and others, that specifically can model the batch effect. So it's very important, especially because this is going out into the internet, as we say, that people keep that in mind. And Jeff Leake and others who were behind that method have made that very clear, that method really needs to be used appropriately.
But to keep his due, I mean, Mark has been really very meticulous about all sources of variation, and this is actually a very good analysis which actually allows you to use surrogate variable analysis.
Right. Okay, well, I think we've got that all recorded for the record. So I want to thank Mark, and now the public will leave and his committee will stay behind, not to be recorded on the record.
Thank you.
Thanks, Bill.
Yeah.
Thank you. Thanks, Nathan.